In this lecture presentation, we'll be discussing cellular communication. As always, I recommend having the slides in front of you so you can take notes as we go, and also to have the interactive study guide available so you can answer the questions as we go through the relevant material. We'll start with terminology related to synapses. A synapse is the gap between neurons. It is the space that neurotransmitters need to cross to impact neighboring cells. We'll talk more about that later in this lecture. Neurons can be categorized as presynaptic or postsynaptic. All neurons can be both. Presynaptic refers to the fact that that neuron is releasing the neurotransmitter or neuropeptide or hormone, that it is delivering transmission. Postsynaptic means that it is the receiver of that transmission. Graded potentials. Graded potentials degrade much more quickly than action potentials. Graded potentials can be subdivided into excitatory postsynaptic potentials and inhibitory postsynaptic potentials. As we talked about in the last lecture, excitatory postsynaptic potentials refers to the fact that the neuron is becoming more positive on the inside of the cell. That means it is getting increasingly closer to triggering an action potential. EPSPs occur because sodium, a positive ion, enters the cell. In contrast, inhibitory postsynaptic potentials hyperpolarize the cell, meaning it becomes more negative in the inside. And because of that, it gets further and further away from triggering an action potential. IPSPs occur because the neuron selectively opens gates for potassium to leave the cell or for chloride to enter. Potassium is positive, so leaving the cell leads to it becoming more negative on the inside. Similarly, chloride, which is negative, makes the cell more negative on the inside when it enters. IPSPs essentially serve as the brake that suppresses excitation for that neuron. So EPSPs depolarize the cell, whereas IPSPs hyperpolarize the cell. EPSPs and IPSPs also affect the neuron's spontaneous firing rate. All neurons can have action potentials without synaptic input. If it's an EPSP, there's increased frequency of spontaneous firing. In the case of an IPSP, there's decreased frequency of spontaneous firing. The next two terms we're going to discuss on this slide are temporal summation and spatial summation. Temporal summation, like it sounds, relates to time. Temporal summation is when many EPSPs occur close together in time. That brings it closer to an action potential. Those signals are summed. Similarly, spatial summation involves summing as well, but in this case, it is many EPSPs that are at two close by locations at the same time. So temporal summation are in the same place over and over again really quickly in time. Spatial summation are at the same time, but at two close by places. Temporal and spatial summation ordinarily occur together, and it helps produce synchronized responses. Let's move on to synaptic transmission. What you see on the slide are the steps from an action potential to the release of neurotransmitters, and then the deactivation of that neurotransmitter. Let's start with step one. So in step one, the neuron has to synthesize neurotransmitters so that they're available for release. Small neurotransmitters are created in the axon terminal itself, whereas larger neurotransmitters are created in the soma. Large neurotransmitters are also referred to as neuropeptides. We'll talk more about that in just a bit. You'll see in the picture that there is a presynaptic neuron towards the top that is releasing neurotransmitters to the postsynaptic neuron pictured below it. The green glial cell that is wrapping around the synapse is an astrocyte, and it's shielding the synapse from outside chemicals. These types of glial cells also interact in the communication process. They help with uptake, enzymatic breakdown, and release to neurons. We'll cover those concepts in just a bit as well. So in step 2, the action potential reaches the terminal. That initiates calcium channels to open, which allows calcium to come flooding into the terminal button. This is what triggers what is known as exocytosis, where neurotransmitters are then released into the synaptic cleft. From here, in step 3, the neurotransmitters are able to diffuse across the cleft and bind to receptors on the postsynaptic neuron. This will alter the activity of the postsynaptic neuron. In step 4, neurotransmitters release from their receptors, and you can think of that as unbinding. So they release away from where they were once bound to the receptor. From here, they can undergo a process known as reuptake, where the presynaptic neuron that released the neurotransmitter takes it back. Uptake, where another cell takes it instead of the cell that released it, and that could be a neuron or a glial cell. Or in some cases, it diffuses away, never to be heard from again. In step 6, the postsynaptic cell may send reverse messages, and in this case, what was the postsynaptic cell actually releases neurotransmitters to the presynaptic cell. This slows the release of neurotransmitters from the presynaptic cell, so it serves as a feedback mechanism. This is one of the ways in which marijuana exerts its effect. Many drugs, including medications, work by affecting synaptic transmission in some way. I recommend at this point pausing the recording to view the video's neurotransmission animations. I included 2 videos on the same page because they are very short. It's about 3 minutes combined. But they do a really nice job of presenting a visual of what the synaptic transmission looks like. We're going to move on to neurotransmitters next, so it might make sense to also watch the video Introduction to Neurotransmitters, and then return to the lecture presentation. Neurotransmitters are chemical messengers, and they're nearly all synthesized from amino acids that are obtained from dietary protein. There are many more neurotransmitters than the ones we'll cover in this course. To start, amino acid neurotransmitters. That includes glutamate, GABA, glycine, and aspartate. We'll talk quite a bit about glutamate and GABA in this course. Glutamate is essentially the on switch. It's an extremely prevalent and excitatory neurotransmitter. The yin to glutamate's yang is GABA. GABA is a very prevalent, highly inhibitory neurotransmitter. In a category of its own is the modified amino acid acetylcholine. Acetylcholine plays important roles in learning and memory, and it's implicated as part of the pathophysiology of Alzheimer's disease. Monoamines all contain one amine group, hence the term monoamine. They are modified amino acids. Monoamines are subdivided into 2 major categories, the indoleamines and the catecholamines. Indoleamines include serotonin and histamine, whereas the catecholamines include dopamine, norepinephrine, and epinephrine. You'll notice in the picture to the right that you can see the process of creating the catecholamines. It starts with phenylalanine, which you get through diet. It converts to tyrosine, which converts to dopa, which you may have heard of referred to as L-dopa. Dopa then converts into dopamine, which converts into norepinephrine, which converts into epinephrine. In contrast, serotonin is created from tryptophan, which you also get from your diet. Tryptophan converts into 5-hydroxytryptophan, which you might have heard referred to as 5-HTP, a popular supplement that many people use for mood-related issues. It converts into serotonin, which is oftentimes abbreviated as 5-HT, which stands for 5-hydroxytryptamine. Unlike the monoamines in general that just contain one amine group, catecholamines are a special category because they contain a catechol group and an amine group. Neuropeptides are amino acid chains. They're larger than the majority of the neurotransmitters, but smaller than hormones. Neuropeptides include endorphins that have been attributed to that runner's high, substance P, neuropeptide Y, orexin/hypocretin, brain-derived neurotrophic factor, which is usually referred to as BDNF. We won't talk much about purines and gases, but to cover it quickly, purines include ATP and adenosine. Adenosine is part of the mechanism of action of caffeine. Gases include nitric oxide and carbon monoxide. So how are neurotransmitters stored and released? At the terminal button, neurotransmitters are generally stored in what are known as vesicles. I think of vesicles as looking like little bobas from boba tea, little kind of gelatin globs. An exception to this is nitric oxide, which is released as soon as it's synthesized, because as a gas, it can't be contained within the synaptic vesicle. Vesicles serve a really important function, which is to protect neurotransmitters from degradation by enzymes. Those enzymes are floating around nearby, so if they weren't protected by the vesicle, they would degrade before they could be released. And as I mentioned, the exception here is nitric oxide, which is released immediately after synthesis. The steps for neurotransmitter release and binding start with depolarization reaching the terminal button. That is the action potential. Again, from here, calcium channels that are voltage gated open because the depolarization reaches that area. From there, calcium enters and initiates the process of exocytosis, which then releases the neurotransmitter from the presynaptic cell into that synaptic cleft, that space between the presynaptic cell and the neighboring postsynaptic cell. From here, it diffuses across the cleft to bind to receptors. And we'll talk about what happens next very shortly. As a note, the receiving cell does not have to be a neuron. For example, at the neuromuscular junction, a neuron would release a neurotransmitter to a muscle cell rather than to another neuron. As I mentioned earlier, neuropeptides are larger than neurotransmitters but smaller than hormones. Neuropeptides are also known as neuromodulators. There are many similarities between neuropeptides and neurotransmitters, but there are also really important differences. Neuropeptides are synthesized in the soma, whereas neurotransmitters are synthesized in the presynaptic terminal. Neuropeptides are released mostly by dendrites, but also by the soma and sides of axons. Neurotransmitters are released at the axon terminal. Neuropeptides are released by repeated depolarization. Neurotransmitters are released by a single action potential. Neuropeptides, when they bind to neighboring cells, initiate the release of neuropeptides from those cells. Neurotransmitters, however, don't necessarily have a direct effect on their neighbors. Neuropeptides tend to diffuse to a wide area. Unlike hormones, neuropeptides are localized to the brain. but to a wider area than neurotransmitters. Neurotransmitters don't travel very far. They mostly stay within the area of the presynaptic and adjacent postsynaptic neurons. Neuropeptides have a longer duration of effect than neurotransmitters. Neuropeptides have an average of minutes of effect, whereas neurotransmitters are very fast and generally only have maybe milliseconds to a few seconds worth of impact. Neuropeptides aren't released as often as neurotransmitters, but when they are, they're released in substantial amounts and tend to have substantial effects. Now that we've talked a little bit about how neurotransmitters are released, let's talk about how they're inactivated. First, I want to say that neuropeptides are not inactivated. They always diffuse away. There are three major categories of how neurotransmitters are inactivated: through enzymes, reuptake, and uptake. There are many different types of enzymes that degrade neurotransmitters, and they're specific to that type of neurotransmitter. We'll cover three of them here, starting with monoamine oxidase, MAO. As you might imagine, monoamine oxidase is the enzyme that helps break down monoamines. MAO mostly breaks down monoamines in the terminal to control the amount. So you can see how important it is that they are contained within those synaptic vesicles. If you've heard of MAOIs, which is a class of antidepressants, monoamine oxidase inhibitors, then you're aware of a medication that operates by blocking this enzyme. By blocking the enzyme, it leads to higher levels of the monoamines, particularly in terms of depression, serotonin, norepinephrine, and dopamine. Catechol-O-methyltransferase is specific to the catecholamines. So the catecholamines can be broken down by MAO and by COMT. These are oftentimes referred to as COMT, and there are medications that are COMT inhibitors as well. So catechol-O-methyltransferase is the enzyme that breaks down dopamine, norepinephrine, and epinephrine. Acetylcholinesterase, as you might imagine, is the enzyme that breaks down acetylcholine. Acetylcholinesterase inhibitors are used in the treatment of Alzheimer's disease. Similarly, COMT inhibitors are used in the treatment of Parkinson's disease. Reuptake is the most common process for the inactivation of monoamines. Many of you are probably familiar with reuptake inhibitors. Perhaps you've heard the term SSRI. SSRI stands for Selective Serotonin Reuptake Inhibitors, and it is the most widely used class of antidepressant medications. Reuptake inhibitors block the process I'm about to describe. Reuptake involves a protein transporter that returns the neurotransmitter to the presynaptic cell that released it. It basically serves as a recycling program for neurons. So essentially, if the neuron released serotonin, for example, rather than allowing it to get brought back to the neuron that released it, the SSRI keeps it in the synapse longer by blocking the reuptake process. That leaves serotonin the opportunity to bind to more receptors and to create more change in neighboring cells. As I mentioned earlier, uptake is very similar to reuptake. The difference is that reuptake is when the cell that released it takes it back, whereas uptake is when another cell takes it. Uptake can occur in a neighboring neuron or in a glial cell. Again, it's the same process where the transporter protein takes it to that cell, whether it's the neuron or the glial cell. This is one of the ways where astrocytes participate in communication. Just like enzymes are specific to neurotransmitters, so are transporters. There are transporters for specific types of neurotransmitters, like a serotonin transporter, a norepinephrine transporter, or a dopamine transporter. Earlier, I mentioned the concept of negative feedback. Next, we're going to discuss two types of negative feedback loops that occur from postsynaptic cells. We'll start with autoreceptors. Autoreceptors differ from postsynaptic receptors. Whereas postsynaptic receptors are located on the postsynaptic neuron, autoreceptors are located on the presynaptic neuron. That is, autoreceptors are located on the neuron that releases the neurotransmitter. Autoreceptors are activated by the neurotransmitter that neuron releases. There are two types of autoreceptors: terminal autoreceptors and somatodendritic autoreceptors, and they have slightly different functions. Terminal autoreceptors directly inhibit the release of neurotransmitters. So when they are activated, the terminal autoreceptor actually blocks that cell from releasing any more of that neurotransmitter for some period of time. Somatodendritic autoreceptors exert their effect by slowing the rate of cell firing, which ultimately leads to less release also. The terminal autoreceptor more directly inhibits the cell, whereas the somatodendritic autoreceptor slows the rate of cell firing, which then indirectly reduces release. Somatodendritic autoreceptors can be located on cell bodies or on dendrites. Again, when they're activated, there's an indirect impact that leads to reduced release of neurotransmitter. Retrograde messengers operate differently. Retrograde messengers are released by postsynaptic cells, and they travel backwards in the retrograde direction towards the presynaptic terminal. From there, it inhibits the release of neurotransmitter as well. Ultimately, while they have different mechanisms of action, autoreceptors and retrograde messengers have one important thing in common, which is that they inhibit the release of neurotransmitters either directly or indirectly, and serve as a major part of the negative feedback loop that helps to keep neurotransmitter release under control. At this point, we've only been discussing chemical synapses. I do want to quickly discuss electrical synapses before we move on to the endocrine system and hormones. Electrical synapses are relatively infrequent in the central nervous system compared to chemical synapses. Electrical synapses are faster, and one example is breathing. And again, as we talked about before, breathing is really important that it is synchronized, and electrical synapses are really good for facilitating that. So very few specialized synapses are electrical. And again, the majority are chemical. As I mentioned, they are faster than chemical synapses, and they help with synchrony between cells. In the case of electrical synapses, the membranes directly contact each other. There is no synaptic cleft. That contact is referred to as a gap junction. There are large pores on both sides that allow ions to pass readily from one membrane to another. They remain open at all times, so when they come into contact with each other, an ion release goes directly through those ion channels to the other neuron. Essentially, when it comes to electrical synapses, neurons act as though they are combined. The last thing we'll be discussing in this lecture presentation is the endocrine system. Hormones that are produced and secreted by the endocrine system are released directly into the bloodstream. They act similarly to metabotropic synapses, in that they activate second messengers. Unlike neurotransmitters, hormones can travel far away from their point of manufacture. Your book has a great analogy, which is that neurotransmitters are making a call to an intended person, whereas hormones are more like a radio broadcast to anyone who's tuned into that station. As I mentioned before, neuropeptides are intermediaries between neurotransmitters and hormones. They diffuse within parts of the brain, but not to other parts of the body, which is what hormones do. There are many important glands of the endocrine system, and we're going to cover them on the next slide. But first, I want to point out the picture on the right. You'll notice that there is an anterior lobe and a posterior lobe of the pituitary gland. The posterior pituitary is actually considered part of the brain, not part of the endocrine system, and it's basically part of the hypothalamus, though it is considered its own distinct structure. The hypothalamus produces, but the posterior pituitary releases. In contrast, the anterior pituitary is glandular tissue and does belong to the endocrine system. It produces 6 hormones that we'll cover on the next slide. The anterior pituitary does actually produce its own hormones, but it is under the direction of the hypothalamus. So the hypothalamus is really the conductor of the endocrine system. This slide presents an overview of the endocrine system with some of the most important organs, hormones, and hormone functions. Those of you who like to pause the recording to watch the videos might consider pausing here to watch the video, How Do Your Hormones Work? Alternatively, you might wait until the end of the presentation to watch that video. Let's start with the hypothalamus. The hypothalamus releases quite a few hormones. Ultimately, all of these hormones function to either promote or inhibit the release of hormones from the pituitary gland. It does this by releasing certain hormones that directly affect the pituitary. For example, I'll mention 3 different hormones that the hypothalamus releases that directly affects the anterior pituitary. First, thyrotropin-releasing hormone, which helps to activate the release of thyroid-stimulating hormone from the anterior pituitary. Next, corticotropin-releasing hormone, which helps promote the release of adrenocorticotropic hormone. Another is gonadotropin-releasing hormone, which helps to promote the release of follicle-stimulating hormone and luteinizing hormone. Now that we've discussed some of the hormones the hypothalamus releases to affect the anterior pituitary, let's cover the anterior pituitary hormones. Starting with thyroid-stimulating hormone. Just like it sounds, this hormone helps stimulate the thyroid gland. Adrenocorticotropic hormone increases steroid hormone production by the adrenal gland, which we'll talk about in just a bit. Follicle-stimulating hormone promotes the maturation of the ovum in females and sperm production in males. Luteinizing hormone stimulates ovulation. Prolactin increases milk production and also has some impact on sexual functioning and breast growth. Growth hormone, just like it sounds, is the hormone that facilitates body growth until you are full grown as an adult. The posterior pituitary gland releases 2 important hormones, oxytocin and vasopressin. Oxytocin is involved in uterine contractions, milk release, sexual pleasure, and bonding. Vasopressin raises blood pressure and decreases urine volume. The pineal gland releases that ever-important hormone, melatonin. Melatonin helps blunt the normal alerting signal that is released from the suprachiasmatic nucleus of the hypothalamus. This is a big part of our circadian rhythm. The suprachiasmatic nucleus continues to produce that alerting signal until sufficient melatonin quiets it down. That's what helps facilitate entry into sleep. The adrenal gland is subdivided into 2 parts, the adrenal cortex and the adrenal medulla. Those of you who know a little bit about the anatomy of the brain might know that the cortex is the very outside of the brain. If you know that, that'll help you remember the difference between the adrenal cortex and the adrenal medulla. The adrenal cortex is the outer layers of the adrenal gland, whereas the adrenal medulla is located internally. The adrenal cortex releases aldosterone and cortisol, really important parts of our HPA axis, one of the components of our stress response that we'll learn quite a bit about in this course. Aldosterone reduces the release of salt in the urine. Cortisol, which is one of our primary glucocorticoids, elevates blood sugar and metabolism and is part of our survival mechanism. The adrenal medulla is also part of the stress response, but rather than being part of the HPA axis, it's part of what's known as the sympatho-adrenal medullary response, otherwise known as the SAM response. It's for quick responses to stress, and it releases epinephrine and norepinephrine, colloquially known as the adrenaline rush. That leads to things like increased heart rate, muscle strength, and vasodilation. The pancreas is involved in diabetes, and that's because the pancreas releases two important hormones known as insulin and glucagon. Insulin helps glucose enter cells, and you might have heard of people, especially those with type 1 diabetes, who are insulin dependent. That's because their body does not produce sufficient insulin, so they have to use insulin as a medication. Glucagon helps convert stored fats into blood glucose. The gonads, including the ovaries and the testes, release our sex hormones, estrogen, progestins, and androgens. These help with sexual characteristics after puberty and some of the differences in our bodies that we might call gender-based differences or sex-based differences. The kidneys release renin, which helps regulate blood pressure and contributes to a phenomenon known as hypovolemic thirst. Hypovolemic thirst is thirst that's caused by a loss of blood volume, either from blood loss itself or from vomiting or diarrhea. Fat cells release a hormone known as leptin. Leptin is part of what helps us feel satiated following a meal. It decreases appetite. The thyroid releases many hormones, but two that are particularly important, which are thyroxine, T4, and thiodothyronine, T3. These help regulate body temperature, metabolism, and heart rate. That's it for this lecture presentation. Please be sure to watch the videos, complete the interactive study guide, study with your Quizlet flashcards, and take your Kahoot quiz. And, as always, please reach out if you have any questions, comments, or concerns about the material in this lecture presentation or anything related to the course.